I'm a Dr. Kang and WSU faculty last 27 years, and I'm a faculty in the chemistry department and faculty in School of Molecular Bioscience and chemical engineering and bioengineering school. So I'm also in charge of directorship of the Structural Biology Center, Biomolecular Crystallography Center. So we are helping a lot of faculty in this university or outside of this university to get their molecular structure determined.
So prior to our protein crystallization, we hired this ActaPure system for the purification of the protein, because we need to reach a really high purity, around 95% purity of our protein. So this ActaPure system helps us a lot. And the good thing about this ActaPure system is it can monitor 3 different wavelengths. So we can monitor DNA, protein, and also other cofactors that has a certain wavelength absorbance. And also we have different kinds of columns, so we can separate the protein by their size, by their charge, and maybe the hydrophobicity. So this Acta can meet our different needs for the protein purification.
So we are researching on the purification and characterize the different protein for different purposes. So in the first step, we purify protein with the nickel column that the protein has the his tag, and based on the affinity with the column, we pure the protein. One of instrument we use to characterize protein is the ITC, isothermal calorimetry, that it has the cell, we load the protein in the cell, and the titrant, the ligand load in the titrant, and after that, based on the protocol, the ligand add to the protein and release the heat. The heat measures by this instrument, and after that calculate the affinity of the ligand and the protein. This is multi-angle light scattering instrument. We measure the oligomeric state of protein by this instrument. So inject the protein goes to the column is the size exclusion column. And after that, there are 2 detectors. One detector is the UV detector, measure the concentration of the protein at the 280 nanometer wavelengths. And another detector is a laser or refractometer detectors that depends on the scattering light, measure the molecular weight of the protein. So based on this instrument, we measure what is the native oligomeric state of the protein, monomer, dimer, tetramer, or other native oligomer states.
So I have 2 different research projects in the Kang group. One of them is called PCPC, the other is called EMOR. They're both proteins in bioremediation pathways. The PCPC is part of the polychlorophenyl pathway for degradation, and polychlorophenyls are a byproduct of wood fixation, and so PCPC is a part of that and one step in the removal of the chlorinated phenols. The second protein is called EMOR, and it's part of the EDTA bioremediation pathway, and so it is a repressor protein. So the protein binds to the DNA and stops the production of the proteins, and when it's released, it allows for the proteins to be made through the DNA. And so when we're screening for crystals, we have this fancy robot here, and this allows for 96 different screening conditions to be tried to see if they produce a crystal. And this has really helped in the last 20 years. This used to be all done by hand, but now this takes less than 20 minutes to do. Here at WSU, we're fortunate enough to have our own in-house X-ray diffractometer. And so this allows for either small molecule or protein analysis of crystals. So when we have our crystals, we're able to mount this on a very small loop here and place it on the goniometer. And the goniometer allows for a rotation around all axes of the crystal. You can either shoot copper or molybdenum at the crystal and collect a pattern of diffraction. And so the diffraction pattern then gives intensity as well and allows for the 3D structure to be obtained with a Fourier transformation.
So one of our crucial steps in our lab's research is solving the protein or enzyme's crystal structure. And by analyzing the protein or enzyme substrate specificity or affinity, we can design or engineer the protein to be a better one or a desired one. So for example, we have a protein structure here, or we have a protein structure crystal here by using the X-ray diffraction. So the beam come from here, and then we will have a diffraction pattern. And after that, we can use that data to solve this protein structure. And this is like a 3D picture of the protein structure, or you can see this is like a real protein structure.